In this video, we are going to discuss about the electrochemical sensors, that is pH. So, by the end of this topic, the students can able to understand the operational mechanics of ion selective electrodes, and also they can able to explain the chemical kinetics of the hydrogen ion diffusion across the specialized membranes, able to apply the Nernst equation to track the potential differences and evaluate the different structural types of membranes. So, pH electrode. pH electrode is by the far the most well known and the simplest electrochemical sensor available. Actually, this is used for illustrating the basic operating principles of ion selective electrodes, or we call it as ISCs. Actually, this is a device. It is a device which is designed for measuring the precise concentration of hydrogen ions in the target solution, and this indicates the degree of acidicity, because pH is mathematically defined as the negative logarithm of the hydrogen ion concentration, and also the pH value of 7 will translate to the exact concentration of 1 into 10 power minus 7 moles. And the most essential structural component of the pH electrode is sensitive glass membrane. So this membrane is uniquely used for permitting the hydrogen ions while blocking all other competing ionic species. When we immerse the electrode into the liquid test sample, the external hydrogen ions will spontaneously diffuse through the glass membrane, and this migration will continues until a state of thermodynamic equilibrium is reached between the external and the internal ionic concentration. So here, as a result, a localized charge that builds upon this inside the walled membrane will be producing a charge which is directly proportional to the total number of hydrogen ions. And the types of membranes. So the membrane is considered to be any material that separates two solutions, because this system must maintain strict equilibrium conditions to prevent the altering the sample. There is very little actual current flow permitted through the circuit. So this delegate potential difference cannot be measured in isolation. It can only be detected through a separate high precision reference electrode. So to record this potential difference accurately without draining the charge, we must utilize a highly sensitive high input impedance multi-volt meter or digital acquisition system. And the exact logarithmic relationship between the ionic concentration or chemical activity is actually governed by the Nernst equation. So it is given here, and here the standard pH electrode and the specialized ion selective course is discussed. So first, it is contrasted to the high specific pH membrane. Other ion selective membranes will be not entirely immersive to this ions, but they can able to permit the passage of secondary ions present in the test solution. So this can be actually done in the Nicolas-Eastman equations. And the difference between the pH and the other sensors is it lies only on the measurement precision value. That is, mathematical calculation is far more dependent on the potential measurements than the pH value. But the pH measurements will naturally care about the general order of concentration, magnitude concentration, and it will be having the minor shift of almost 0.1 pH volts. And the types of membranes, to understand how we minimize this interference, we must study the structural type of membranes. So in electrochemistry, a membrane is actually defined as a material layer where it separates two distinct solutions. So it is directly across the specific material boundary. So today, several types of sensing electrodes are commercially available to clinicians and researchers. They are strictly categorized by the nature of the membrane material they are using for constructing the electrode bulb, and this is the exact variation in the material fabrication where that makes the electrode selective for one specific ion over another. And the next is the glass membrane. So our first major category is the glass membrane. So these membranes are manufactured from a specified ion exchange type of glass that is usually consisting of silicate or components, where here you can see the excellent selectivity material that single charge cations can be extended to the selectivity of the certain double charged metal ions also. So this is the universal example of the quantity in clinical pH glass electrode. And exact physical assembly of this pH glass electrode is manufactured from the silicon dioxide network. So this can be characterized by negatively charged oxygen atoms, and when the entire electrode assembly is immersed into the liquid sample, there will be an electrical potential across the glass bulb which will allow us to read out the pH value directly through the high impedance data acquisition measurements. And in crystal membrane electrodes, our second major category is the crystal membrane electrodes. So instead of an amorphous glass network, these membranes are fabricated using mono or polycrystals, and also these offer a particular selectivity because the physical dimensions of the crystal lattice structures are highly rigid. So this is the best example for the commercial fluoride electrode, where we are using in the lanthanum fluoride crystal matrix. Thank you.